Excited to present our next speaker, Prof Grienberger. She is also a medical doctor by training and holds a PhD in neuroscience from the Technical University of Munich. Prior to joining Brandeis, she was a postdoctoral researcher in Jeff McGee's lab at Janelia and then Baylor College of Medicine in Houston. Her research bridges cellular and systems neuroscience and focuses on the synaptic, cellular, and circuit mechanisms underlying experience-dependent neural computations in the brain and the disruption of these computations in animal models of Alzheimer's disease. Her talk is entitled Dendritic Computations Underlying Experience-Dependent Hippocampal Representation. So join me in welcoming Prof Grienberger.
Thanks, Jasmine. Thanks, Dan, and really thank you all the organizers for having me. It's my first time at the meeting. I've learned a lot, and I'm looking forward to the rest of the meeting. So I'm an experimentalist by training, and my lab combines experiments in animals with computational methods to sort of try to figure out what are the mechanisms that allow our brains to learn. The core notion behind the work that I'm going to present today is that learning requires a set of experiences, and these experiences somehow drive changes in brain activity that are persistent and are really required for animals, for really any individual to learn tasks that are important for their survival. And in a mouse, and that's the model we are using, this could be remembering where to find food, where to find shelter from a predator, etc. And so despite this important function, I would argue that that how the brain accomplishes this is really not well understood. A lot of seminal work has been done in reduced brain preparations, such as culture and slice, but how it works in the intact brain is really not well understood. And so I'm going to talk today about our efforts to try to understand these processes better, with a specific focus on dendrites. These are these beautiful tree-like structures where really neurons receive most of their synaptic input and computations performed by these dendrites. And I hope by the end of the talk it will be pretty clear what I mean by computations in dendrites. And before I start, I should really acknowledge that this was work that was started when I was still a postdoc in Jeff McGee's lab, and then it was finished in my own lab as Brandeis. Okay, so to get at that question, how experiences change brain activity, how really experiences are transformed into memories, if you will, we have been studying place cell activity in the mouse hippocampal area, CA1. And CA1 is, of course, a good area to study this sort of questions because we know from really decades of research at this point that the hippocampus and intact hippocampus is really necessary for performing all sorts of learning. And so let me introduce briefly what place cell activity is. So place cells are neurons that we can find in the mouse and really in many different species that are active when the animal is in a particular location of this environment. And here I'm showing you one example from one place cell. The white line corresponds to the trajectory of the animal as it explores an open field arena, and the red dots are the action potentials, are the activity that this neuron have. And so you see this place field of this neuron is in the right upper corner of this open field arena. Now, in any given environment, about 25 to 50% of CA1 neurons are place cells. Their firing fields, or place fields as they're called, tile the entire environment. And so when John O'Keefe discovered these cells, the first idea was that these are sort of this activity, these representations formed by places are really related to how animals navigate in space. However, when one looks at the population activity, and this is now data from my own recordings, and so these are around 1,200 place cells or so. Each row is one place cell. The x-axis shows the position of the animal on a linear track, and the red color marks really for all of these place cells the center of its firing field. You see that, yes, that the firing fields tile the entire environment, but there's also a location at 90 centimeters where you have a clearly an increased place field density. And this is the location where the animals that are water restricted receive a water reward. And what this suggests to us that actually what this place cell representation in the hippocampus reflects is kind of what is important, what is behaviorally salient to the animal. It reflects, in a sense, the experience of the animal. And so we decided to use studying how this representation form as kind of a model system to study how experiences affect brain activity. So let me introduce quickly the anatomy of CA1. So this is an individual CA1 pyramidal cell neuron. You have the soma and the axon at the bottom. And then really each pyramidal cell anywhere in the cortex, but also in CA1, has these two dendritic integrative compartments. So you have the dendrites that are close to the soma. They receive local input. In the case of the CA1, that would be area CA3. And then there are these very distal dendrites called the tough dendrites, which receive a different input from a different brain region. In the case of the CA1, that's layer 3 of the entorhinal cortex. And so the biophysics of the cell dictates that really these compartments are really quite integrating these inputs separately from each other. In addition to these excitatory inputs, we have also a sort of a set of inhibitory inputs. And so what my interest is and what this project that I'm going to talk to you about today is about is how do cells integrate these inputs to make sense of them, to create something that's a place cell output, that's presumably a behaviorally relevant action potential output. What is the role of synaptic plasticity and other properties, such as, for example, excitability changes for this process? So the main question is, how do cells become place cells? The task that we're using, so as I said, we're recording in behaving animals. The task is very simple. The animal is head fixed on a linear treadmill. The treadmill is 180 centimeters long, and it's 3 sets of somatosensory cues attached to it, which sort of mark the different locations on the belt. And the task is very simple. The animal has to learn a fixed reward location on this belt. And they are, since the animals are water restricted, it's very easy to train them. And within a session of usually 60 minutes, they become very good at it. And so this is one example here. You see this is the real life. So this you see here the animal, it's head fixed, it has the lick port attached to it. You can see the belt passing by, and you can hopefully get an impression about the somatosensory cues that are attached to the belt. And so below you have a readout of sort of the behavioral parameters we're analyzing. Usually that would be velocity, that would be the licking specificity, and that would be the running pattern and the distance that the animal runs. And so in a very well-trained animal like this one, you see that the animal sort of restricts its licking to a tighter and tighter zone around the reward in red. And it also has this stereotypical running pattern where the animal will slow down as it approaches the reward, consumes the reward, and then accelerates again. Okay, so combining this task, then we found a few years back that a specific type of burst firing, and these are intracellular in vivo whole cell recordings, so that a specific type of burst firing is a very powerful driver of de novo place field formation. So in this cell, you see that in labs 5 or 6, so this is a recording from one cell, in labs 5 or 6, and I'm having the numbers here on the left side. So in labs 5 or 6, the cell was not very active. It only fired a few spurious action potentials. But then after firing one of these bursts that we call plateau, because they reflect that this is the somatic signature of a specific dendritic event, a dendritic plateau potential. So after the animals fired this specific event, you see that a place field would form and would stay there for the remainder of the recording. Now, through a series of analyses and other experiments that I would be happy to address in the discussion if there are questions, we basically concluded that what is going on here is synaptic plasticity. Basically, certain synapses changing their weights, which would then convert the cell from a silent cell into a place cell. We call this plasticity behavioral timescale synaptic plasticity, or BTSP. And so this is how it works. So you have a neuron, you have after it fires a single dendritic plateau potential, you have then synaptic weight changes, specifically not on the tuft inputs, but on these local perisomatic inputs. And this, the plasticity kernel is on the behavioral time scale. So you have these synaptic weight changes of inputs that can start up to 3 seconds, 4 seconds before the plateau and last for up to 1 second after. So it operates really on the seconds range, hence the name behavioral time scale. I also want to point out, because of the discussion yesterday about the successor representation, that this is an asymmetric plasticity kernel, as you can see, right? It lasts much longer forward than after. And so the consequence of this asymmetry is that actually the place fields that are generated by PTSP precede the actual plasticity-inducing event. So this is shown here in one cell, but actually on the average in our population of 20 cells or so for this paper, the shift in forward was around 10 to 20 centimeters. And so we call this predictive place cells. So that means what we want to say is that basically the firing that was generated precedes the actual event that drove the change in to place cells. All right, so back to this original question. So is BTSP? It's one plateau is enough. It works on the behavioral time scale. So is then BTSP the mechanism that allows actually representations, that allows experiences to be converted into neural activity, if you will? And of course, plateaus would be sort of the major player here. They would be kind of a teaching signal telling the cells, okay, here something significant has happened. Now form a place field in this location. So to get at that question, we turned to a different technique. So the whole cell recordings are great, but you can only record a single cell at the same time. So you need a method that allows you to record many cells at the same time. And so this is a very classical neuroscience technique by now. So it's called two-photon calcium imaging. So we can then use a microscope to record hundreds, maybe even thousands of cells at the same time. So on the video on the right upper, you see sort of these donuts. They are filled with a calcium indicator, and whenever a cell lights up, basically we can assume that it was active. And so this allows us then to record the place field activity of many cells at the same time. And we can do this, as you can see, in behaving animals. It's pretty standard at this point. So the question is basically, what's the connection between the single cell mechanism that we identified, BTSP, and this experience-dependent representation with the reward over representation that I showed you at the very beginning? So is there any connection? So to get at that question, we wanted to look at how do individual place fields form in the calcium imaging data. So I'm showing you here 3 different examples of cells. The blue, this is now each on the y-axis you have individual labs. On the x-axis you have position on the linear track. And so you can see that all these 3 place cells were not there from lab 1, right? They all sort of arrived later in the session. And importantly, they kind of went from there was no activity in the cells, that would be the color blue, to high activity, which would be the color red. So this is already very reminiscent of this one trial learning rule, BTSP. The other feature that I want to sort of point out is this predictive shift again that I mentioned. You can see that all these place fields and the induction lab, the lab with the plateau is marked in red in this case, you can see that all these place fields sort of shifted consistently forward. Again, this is a consequence of the asymmetry of the BTSP plasticity kernel. And so we can already conclude here that most of the cells kind of displayed signatures that would be characteristic of BTSP. And I'm not showing here the population data because this is published, but this holds also true for on the population level. I'm going to show you some more data. So we also can show that the formation of this representation is sensitive to pharmacological blockade of BTSP. So we used an NMDA receptor antagonist, AP5, and a calcium plateau blocker, SNX. And in both cases, 2 things happened. One, the overall number of place cells went down. Usually it's around 25 to 30%. In this case, it was 16% or 12%. And also sort of the reward over representation went down. So you can see that in comparison to the control, where you have a lot of place fields around the reward marked with a dashed line, you now have a more uniform distribution. So this is sort of all consistent with then the following idea. So CA2 has these two different inputs coming from CA3 and EC3. You have BTSP on these CA3 to CA1 inputs, and this creates then an experience-dependent and predictive representation in CA1. Now, the last question was, why is this happening? Because what we would have to assume is that there is more BTSP around the reward, hence the reward overrepresentation. And so that means that something must drive these plateaus at this location. So the assumption was that maybe EC3 is sort of the driver for these plateau potentials that then would cause BTSP. So what I did is I sort of optogenetically silenced these EC3 inputs, and this indeed led to a decrease in the place field formation on this location. So this was the first check mark that was, it showed that EC3 was indeed necessary for this BTSP-mediated place field formation. But then we also wanted to understand what is the nature of this input. How does the EC3 do that? And so to get at that, we sort of turned to calcium imaging again, but now we looked at the activity of EC3 neurons, which we can access through the axons of these neurons in CA1. And so these are 3 individual axons. You can see that they are all, so on the top you have again the heat maps, labs are on the y-axis, position is on the x-axis, and below you have sort of an average collapsed across all these labs. And you see that when you compare it to the place fields that I showed you before, there are some striking differences. One is that they are not as selective, individual axons are not as selective as the place cells, and the other, it's not as reliable, the activity. The activity is much more stochastic, if you will. Right, so it's not every lab will have a reliable activity burst. So we wanted to quantify that a little more, so I split up the data into 2 parts, odd labs, even labs. And you see now 2 heat maps. The even labs, the averages of the even labs are sorted according to the odd labs, and you can see sort of this messiness show up, right? For example, you see that if these cells would be really reliable, you would see sort of the same same diagonal in both plots, but that's not the case at all. So to kind of get at it a little more, I'm now plotting for each of the axon, the peak location in the odd and the even labs. Each dot is 1 axon. And again, it kind of shows sort of the stochasticity, as well as that there are some axon on the unity line, meaning those are really the correlated ones, but they are far in between. It's maybe 25% of the cells. So, and even if we look at these really correlated ones, it's not really particular for the reward location, which would be here at 90 centimeters again. So this was all quite disappointing because sort of the normal hypothesis was maybe there is just more input at the reward, hence more plateaus, hence more BTSP, hence more place field formation. But that was clearly not the case. So what we instead had to conclude is that maybe what this activity reflects in the end on the population level is sort of the uniform Q distribution. I showed you before that sort of the Qs on the belt are everywhere. So there's not a specific a higher density somewhere on the belt. And I will get back to this point later. So to understand, though, how this input might be driving plateaus, we then turned to modeling. We made a very simple Markov chain model where we modeled 2,000 EC3 axons as Markov chains. These Markov chains recapitulate really well sort of the biologically observed pattern. And then these 2,000 axons impinge onto 10,000 downstream CA1 neurons. We randomly, for each of the 10,000 neurons, we randomly select 200 axons. We sum their activity up. And then we say this is the input that this neuron gets. And what we can see is that sometimes, and I'm showing you here 5 examples, axons, sometimes if you sum this sort of stochastic, spatially unreliable activity up, you will have a large activity peak, which will cross a threshold. And maybe these are the events that will drive plateaus. So you have sparse events, sparse times when the CA1 gets higher input, and this could be when the plateaus are induced. So we can then ask, what's the spatial distribution of these threshold crossing events? We can have two cases. One is where the running pattern is uniform. That's here plotted as the dwell time. So that's just the inverse of the velocity. So if we have a uniform dwell time, the spatial distribution will not be different. It will be everywhere the same. But if we then impose the actual running pattern where the animal slows down when it receives the reward, the dwell time will be higher. Then we can see that now there is an increased probability of seeing threshold crossing, plateau evoking events there. And so what we concluded from this is that this E33 activity is consistent with an instructive signal driving BTSP, but there is two aspects to it. It's the pattern of the activity, but it's also taking into account the behavior. Now let me get back to that notion that, so this is the summary, sort of in this environment A, where we have cues on the belt. You see that basically the EC3 instructive signal is kind of has a peak and the CA1 follows it. But the notion was maybe, what about the cue distribution? Does this matter? And so I did a second environment where I had only two cues now, one visual stimulus and one reward. And you can see that in this case now, the instructive signal is quite different. It does not is not higher anymore around the reward, marked with a white dashed line, but it's now higher right after the visual stimulus, which is marked with the blue line. And so then, if we look then at the CA1 representation, you can basically see that also in this case, CA1 follows it. So now we don't have a reward overrepresentation anymore. Now we have an overrepresentation of the light. Okay, so last point. Is this instructive signal an error or a target signal? The difference would be, the idea would be that an error goes away with learning, a target stays constant and does not change with learning. So here's our perhaps naive attempt to get at that. So I'm showing you here in red just the summed up population activity of CA1 for labs 1 to 10 and labs 51 to 60. So this is when most of the learning happens, when most of the animals learn where the reward location is. You can see that there's an increase in the overall activity, and there's an especially peak around the light in this case, because this is data from the light environment at around 40 centimeters. If I now plot on top the EC3 activity, you can see that it doesn't change. It stays constant. And so we can quantify that now for 80 labs, and we can see that basically the correlation between the red and the blue line gets closer and closer to each other. So this is sort of the summary of my entire talk. So this is sort of the working model that we have now for this BTSP mechanism. So EC3 has an instructive signal. It's a target. It's a stable signal throughout learning. You have then in the CA1 dendrites a mismatch calculation between this target and an as of now unknown signal that signals the state of the actual CA1 representation. If there's a mismatch between the two, so if CA1 has not adjusted, there will be a plateau fired. BTSP will change the representation, and then the cycle can begin from the beginning. And so what this then allows us to do is, I think, two significant things. One is we propose here for the first time a plasticity mechanism that allows to coordinate plasticity across connected circuits. So it's not only a local phenomenon, it actually allows us to coordinate plasticity. And the other is that it provides a potential answer to what is known as the temporal credit assignment problem. How can actually a delayed outcome and the synapses that led to that delayed outcome be connected? And so this mechanism allows for both of these provides answers to both of these problems. Many outstanding questions. What is the role of neuromodulation? How is it in other brain areas? Is it something really general, or is it just the hippocampus? And of course, most importantly, how does the EC3 know that it should form this instructive signal? As I mentioned, this work was started in Jeff McGee's lab. I now have my own lab at Brandeis. It's really a privilege to work with these fantastic group of people. Thanks to the funding sources, and thank you all for listening.
Hi, great talk. So for the target signal, sorry, I'm here.
Yeah.
For the target signal coming from EC3, is that related to licking, or let's say, so what would, is it the motor action itself? Is it the expectation of water? What would happen if basically there is no water when they're licking?
Yes, I would not say it's the motor signal per se, right? Because, for example, in the second environment, actually the signal is high around the light and there is no licking going on. There's also not a big change in motor behavior at this point. So what the EC3 gets is some sort of multimodal sensory input from the rest of the brain. How this is then, calculated is really an unknown at this point. Not much is known about how individual EC3 neurons process their inputs. Yeah, I don't know. But I think what hopefully was clear is that we have these two different environments, and the EC3 signal changes somehow, and then CA1 follows it. So this is as far as we can say at this point. Yes.
Thank you for the talk. It was a very nice talk. So I have a question about the plasticity. So you mentioned that plateau potentials trigger plasticity in the inputs from CA3, but not from entorhinal cortex. So do you think these inputs are not plastic? They are not changed by plateau potentials?
No, I do not want to say that at all. And we know from slice recordings that they are. That's actually something we're actively looking at right now. It's possible that there is some sort of storing from day to day going on also of the signal, right, of the instructive signal. So that could be how cells from day to day, keep their feature selectivity. I mean, I'm just trying to be careful because we really have only solid evidence for the CA3 to CA1 input at the moment. But we know from, LTP slice recordings, so not in the BTSP paradigm, that these inputs can undergo plasticity.
And I have also another question.
Sure.
In your talk, you mentioned only potentiation of inputs from CA3. So are there conditions in which these inputs can depress?
Yeah. There is actually, I don't know if we have time. I mean, we can talk offline. So there is, in a follow-up paper, which I just left out right now, we show basically there is also depotentiation going on at the same time. So there is, in each cell, there is always a balance going on between potentiation and depotentiation. And the punchline is that it basically depends on the weight of the synapse when the plateau happens. High synapses with higher weight that are further away from the plateau will more likely undergo depotentiation, whereas low weight synapses will undergo potentiation. I can point you to the paper. It's an eLife paper that came out in 2021.
Thanks. Do we have time for one more? Thanks for an excellent talk. I'm just curious what happens with these EC3 cells if you just abruptly remove the reward and stop giving it.
Oh, what would happen? In the behavior, you mean, or?
No, in the cells. Just because there's this beautiful one-trial learning, I'm just curious about extinction.
So what has been? So I haven't tried this, really consistently. Just have anecdotes. So what basically happens? The whole representation disappears, and there was a beautiful paper from Chris Harvey that basically showed that task engagement is quite important in the whole system. So basically, when you remove the reward, I think it's not so much that these animals lose their spatial representation per se. It's just that the task engagement goes away, and so the whole system basically, the whole hippocampal system loses their selectivity in a way.
Thank you, Christine.